Students, today I would like to discuss about the various apparatus, equipments and the small instruments that are used in the microbiology laboratory. Now every one of us know that all laboratories have certain specific instruments that are entirely or specifically required for that particular laboratory. Similarly, microbiology laboratory has some apparatus, equipments and instruments apart from the routine apparatus that are present in all laboratories. For example, if we consider that the glass apparatus like conical flask, the beaker, the measuring cylinder, the pipettes, these are the common glass rods, these are the common apparatus that are used in all laboratories. But apart from this, there are certain specific equipments in the laboratory of microbiology. When we start with the laboratory of microbiology or any laboratory, we come to the word safety measure of the laboratory. And here in this microbiology lab, apart from the fire safety and the water safety, there is another word called contamination free zone. We maintain the sterility of this laboratory to a greater extent. For this reason, the first thing when we enter the laboratory of microbiology, we should keep into mind that the laboratory should be free from all outside environment dust particles and other microorganisms that are not required for the study. For this reason, while entering the laboratory, we should wear an apron, a mask, the gloves, and possibly the hair cover or the cap. We should change the shoes that we are wearing outside, and either we should wear a slipper or we should wear the shoe covers. It should be totally contamination free, and this reason is that the microorganisms, they change or they die if the growing environment changes. Similarly, if the microorganisms are not properly treated, they may enter our body who are doing the experiment, and we will get a new or we will develop a new disease. The second thing is that we should wash our hands thoroughly with the soap before and after the experiment to prevent any type of contamination. Now coming to the main topic, that is the various apparatus. As we had said that all materials that we are using or all apparatus and the equipments we are using, it should be in the sterile condition or it should be a clean one without any type of contamination. For this reason, what we should do is we are supposed to take those materials that can be cleaned very easily and that can be sterilized. I know that you people are very much aware of the word sterilization. Now this sterilization means that we should keep all apparatus and all equipments away from any contamination. For this reason, the most important material that are used for the glass apparatus for the microbiology lab is the glass, because two things are very important for the glass. The glass is very inert, it does not react with any type of substances, and secondly, the cleaning and sterilization of the glass is very much easy. For example, if we go for sterilization, we require heat in maximum cases, and the heat, it is always a heat protectant, or it can resist, heat resistant. I will say that it can resist the heat. Now coming to the basic equipments or the apparatus that are made up of glass, we know that there are beakers, measuring cylinders, the glass rods, these are the common. Then what is important for the microbiology laboratory? The first comes are the test tubes. Now test tube, if you see, there are two different types of test tube. This is one and this is the other. Here I have separated another one. What are these? One is with rim, the other is without rim. Why with rim and without rim? With rim we know that for dilution, for addition of reagent, we use with rim. And without rim test tubes, they are also called the culture tubes, which are used to prepare the various slants of the microorganisms. Slants, we know that they are the solid medium that is prepared in the test tube in a slanting position where we allow the microorganisms, especially the bacteria, to grow over them. Now these are the two main types of test tube. Apart from this, there is a small tube you see, the name is Durham's tube. What is Durham's tube? It is of around 5 to 6 centimeter in length and a very narrow tube. We also call it as the fermentation tube, and here we check, this tube is used to check the fermentation of a microorganism. Suppose any microorganism undergo fermentation reaction. If we grow it in the liquid culture media, that is broth, what we do is we take the Durham's tube and place it in the inverted manner into the test tube. What will happen after 24 hours or 48 hours of incubation? We see that gas is there on the top of the Durham's tube. The gas layer indicates that the fermentation process has taken place and there was a gas formation. Now, the next slide we come, the Petri dish. What are Petri dish? These are the glass plates, you know. Here you see, these are the glass plates. It has two parts, the lower dish and the covering upper dish. What actually is done is a solid agar plate is done, that is a solid medium. Especially the test tube, we can do the liquid medium. When solid can be done, but in the plate, never a liquid medium can be used because it will fall. It is usually the solid medium that is done over here, and what is done, that is usually streaking, that is to separate the microorganisms, the colonies of bacteria. We can count the bacterial colonies over here. We can do the antibiotic assay in these plates, and also we can grow the microorganisms in the plate. So this is the Petri dish. The second one is the two you see over here. One is the McCartney's bottle and the other is the Bijou bottle. The McCartney and the Bijou bottle, these are the glass bottles with the aluminium screw cap. The McCartney bottle is used to transfer the bacterial growth in the liquid or the semi-solid medium. And Bijou bottle, they are of two sizes. One is the large Bijou bottle, the small Bijou bottle. One is of 10 ml capacity, the other is of 5 ml capacity. And they are used to culture the media in both the solid, semi-solid as well as in the liquid medium. And we can also grow the microorganisms in the Bijou bottle. That is called culturing or cultivation of the bacteria. The next one, the pipettes. We know that we people are very much aware of the pipettes, and in normal laboratories, the chemistry laboratory or the pharmaceutics laboratory, we have come across the two types of pipette. One is the graduated pipette and the other is the 1 mark pipette. Here also we find the two different types of pipette. One is the micro pipette and the other is the pass-through pipette. Now if you see over here, this is the micro pipette. This micro pipette, we can take the quantity of the material in microliters. And usually nowadays there are many types of micro pipettes, and here the micro pipettes can be of 3 different ranges. One we can find is of 2 microliter to 20 microliter capacity. The other is of 10 microliter to 200 microliter capacity. And the third one we find is of 100 microliter to 1000 microliter capacity. Here, if you see, this is the tip of the micro pipette and at the end, and here what happens, we add the various tips that are available of various capacity, and those are the polyethylene or polypropylene tips. Those are sterilized materials which can be used only once. Then the next comes the pass-through pipette. It is different from other pipettes. there is a rubber cork that is added over there, and you see over here the tip is a long slender. Why it is like that? These are usually used to collect the reagents from the reagent bottle, or the dye which we require it in a very small quantity, as one drop is equivalent to 0.02 ml. The last one is the graduated pipette we use. Graduated pipette means there are graduations. What do we mean by graduations? Graduation means there are marks of the various volume on the pipette. On top you see a case, that is a metal case that is present, and in this metal case the pipettes are kept. This is where the pipettes we keep it, and we take the entire metal case and put it for sterilization. While doing a sterilization, remember the cotton plugs are, means the pipette is plugged or blocked with the cotton plugs over here on the top. Even for the pass-through pipette, we put the cotton plugs. Okay, before I proceed to the next one, let me say that when we use any apparatus, glass apparatus in the microbiology laboratory, the first thing we should keep in mind is the washing should be very clean. No particulate matter should be present in the glass apparatus. For that reason, remember that whenever we take our glass apparatus, we use it. After that, first we should wash it with soap thoroughly. Second, we should wash it with the running water. Then we should rinse with the distilled water. Rinsing with the distilled water, and then it should be sterilized before we use for the second time. Now, here if you see, these are the small equipments, the metal equipments that we use in the microbiology lab. The inoculating loop and the inoculating wire. Now, this inoculating loop or inoculating wire, as the name suggests, helps in inoculation. Now, what is inoculation? Inoculation means in microbiology, the bacteria or the fungus, we need to continue its growth, continue its cultivation. How do we do it? We always transfer the microorganism, that is bacteria or fungus, from one media to the other media. When we transfer it from one media to the other media, then what we do is we take this inoculation rod, inoculating loop or the inoculating wire. A small amount of inoculum. Inoculum means a very small quantity of the microorganism is taken from the medium and is transferred to a new medium where they will start growing. Now here if you see this one, here if you enlarge it, you can see a small loop, a circular hole is made with the wire, and in case of the inoculating wire, there are the straight wires have been kept. Now the other name for inoculating loop is the smear loop, that is for collection of the inoculum and sticking it on the surface of the plate or on the slant or adding it into the liquid broth. And in case of inoculating wire, it is also called the inoculating needle. Now remember here, whenever we take a loop or we take a wire, the length of the wire should be long. Why? Because this part, that is the wire or the loop, this part should not touch with any other surface of the tube or the bottle anywhere. It should only take up the inoculum. This is because during a sterilization, remember our inoculating loop or inoculating wire is always sterilized before and after use by heating it to a red hot condition over the Bunsen burner flame. And while heating it, this entire part cannot be heated. It is only the wire or the loop that can be heated. So no part of the handle, the glass handle, the metal handle can be touched with any part of a test tube or the bottle. Otherwise, we cannot make it contamination free. For this reason, the wire needs to be long. And this wire is made up of the inert material, either platinum or the nichrome wire. Now, when we talk about sterility, it is always necessary that any transfer of material in the microbiology lab should be done with the help of the forceps. Without forceps, if we touch it, there are chances that it gets contaminated. The cork borer. What is the function of cork borer? Now, in case of microbiology, different assays are done. One of them is antibiotic assay. This antibiotic assay, we do it where? We do it on the Petri plate. And we prepare the plate with the help of an agar media. And it is a solid medium. Now there we need to prepare wells. Wells means small deep holes. And these holes are made with the cork borer. They form a deep well or a deep hole where the antibiotic is given. The microorganism is already present in the media, and it will indicate that this antibiotic, how much it can inhibit the growth of the microorganism around that hole. This is used, so your cork borer is used to prepare the hole. The next one is the digital pH meter. We know that digital pH meter is present in every laboratory, and microbiology requires it very efficiently because pH plays a major role in the culture media and in the various solutions or buffer solutions we prepare in the laboratory, because microbiology means in microbiology the bacterial, the fungal, the microorganisms rather are very much sensitive to the pH. A small change in the pH will stop its growth or lead to the death of the microorganism. So whatever we prepare, the pH should be checked and it should be maintained as directed. Now remember one thing, this pH meter should be taken a special care, because otherwise the electrode may get spoilt and we cannot use it later on. The electrode is made up of silver coated with silver chloride, and when not in use, it should always be dipped in the concentrated saturated solution of potassium chloride with pH 7. Now before and after use, the electrode should be always washed with the distilled water, and it should be dried with the tissue paper and then dipped with the potassium chloride solution. The weighing balance is important, and again because the media, the minerals, the various powders, the various salts that we take to prepare a medium should be weighed and given in proper quantity. Otherwise, a change in the concentration of the substances in the media may lead to the death of the microorganism, or will retard the growth of the microorganism, or sometimes it may lead to the change in the phenotype as well as genotype change in the microorganisms, and as we call mutation of the strain may occur and we may not get the desired effect. Then the next comes microscope. As the name itself suggests, this is the prime requirement of the microbiology lab, and in this there are different types of microscopes that are available, which we may discuss later on, because microscope itself is a big chapter. We should know elaborately what about microscope, and these microscopes are used to see the microorganisms, the colonies of microorganism under it, and we can, especially in gram staining, the basic requirement in a microbiology lab of an institute, it indicates that whether it is gram positive, gram negative, and secondly we can also understand whether it is a rod, whether it is a cocci, that can be easily detected by the microscopes. And remember, when we use a microscope, suppose the lens is not proper, we should always wash, or not wash, we should always clean the lens with the help of acetone. As we had said, all substances, all materials, everything should be sterile. Secondly, when we take any apparatus, especially the small glass apparatus, it should be properly cleaned. For that, ultrasonicator is an instrument that is used in the microbiology laboratory. Now, ultrasonicator has various functions, but in case of microbiology laboratory, the ultrasonicator is used to clean the small bottles like the McCartney bottle, the Bijou bottle, then the Durham's tube. What happens? Sometimes the small deposit remains over there even after thorough cleaning, or we may not pick out the matter that has got attached to the surface over there. So what we do is, here you can see the bath. In this bath, the water is added, and to that water all the apparatus have been given. And now what happens if you switch it on? The high frequency electrical energy is converted into ultrasound waves. There is a presence of ultrasonic transducer that converts it. And there is the formation of a vacuum bubbles. These vacuum bubbles are very small. They are minute particles, and what they do, due to the presence of the ultrasonic waves, they will expand and burst, and this is called cavitation process. Now there is numerous formation of this bubbles, and they burst continuously. They will expand and burst, and they will act like brushes. So when it comes in contact with that area where the surface has been deposited with the material, continuous cavitation will lead to the cleaning up process. Now in this microbiology, the next is the colony counter, and this colony counter, we normally, if we see a plate of a plate under a microscope, or we cannot see a plate under a microscope, sorry, if we see a plate, and we will see the various cultures are growing, and these cultures are in colony. We cannot see a single bacteria under the naked eye. So what we see is the colony of the bacteria. Now to know how much colony is growing, or what is the optimum amount of colony grown in that media, the best way of detecting it is the colony counter. Here it is an illuminated surface, and the magnifying glass is there. What we place the plate over here, the Petri dish over here containing the agar plate, and we can count the number of colonies that are available with the help of a pointer pen. If we touch those pointer pen, then automatically, digitally, the recordings will be shown, and we can say that how many colonies are there. Sometimes it happens that to maintain the sterility, we cannot use the sterilization process of adding chemicals, or adding the or heating it or sterilizing in the hot air oven or in the autoclave, that is, heat is required. So because there are many thermolabile substances are there. So what usually is done, in a large volume, in a bulk volume, the membrane filtration apparatus is used. This membrane filtration apparatus, it has a cup on top and a conical flask of a large volume is present. For example, 2000 ml, the conical flask is present. We pour the substance over here, the liquid over there, and while pouring it, it will pass through the membrane. Now, when it passes through the membrane, then what will happen? This membrane has a pore size of 0.45 micron. So, any microorganism of larger size than 0.45 micron will not pass through this membrane. We can use it for two ways. One is that we can sterile a bulk solution which is thermolabile using this membrane filtration apparatus, and the second thing what we can do is, if you want to check that whether any microorganism has grown in that pharmaceutical product or any liquids, what we do is, we take up this membrane filter, the filter we take it up, we put it on the Petri dish having the culture media, we allow it to incubate for 24 hours or 48 hours, and we check that whether there is any microbial growth or not. If there is a microbial growth, that means the liquid or the preparation has the microorganisms in it. Antibiotic zone reader is similar to that of a colony counter. In colony counter, we were studying that what was the number of colony formed, and in antibiotic zone reader, what I already said, in the Petri dish we are making the hole, that is the whale, with the help of a cork borer, and then we are adding the antibiotic to it. The media already contains the microorganism. Now, after 24 hours or 48 hours of incubation, we will just check that how much area around that well has no microbial growth. And this is the zone of inhibition. And this zone of inhibition indicates the effectiveness of the antibiotic against that particular species of microorganism. And to read that zone, we can use the antibiotic zone reader. Last but not the least, the most important of all the equipment is a Bunsen burner, because Bunsen burner is required for heating each and every material of the equipment in the microbiology laboratory. For example, I was telling about loop, the inoculating loop. Before we use it, after we use it, we should heat it to red hot. The wire should be heated to red hot condition. Secondly, for example, we are opening any test tube. We should just touch it in front of, or hold it in front of the flame, so that any microorganism or any contaminant, if present, will be killed in that heat. Now this Bunsen burner should remember, whatever Bunsen burner we use, when the flame we use should be the blue flame, that is the heating flame. No yellow flame is allowed. Now this blue flame is the hottest flame of a Bunsen burner, where total combustion of the air has taken place. And to get a blue flame, the collar should always be open. That means the collar should not cover the hole. The hole should be open so that total combustion takes place. So this is all about the preliminary equipments and the apparatus that are used in the microbiology laboratory. Now you can go through these references for further details, and thank you, students.